Cell cycle, cell division. Phases, mitosis. Phase when the actual cell division or mitosis occur. Apparent phase or division phase. Phase between two successive M phases. Interphase. Phase between two successive M phases. Interphase. Phase lasts more than 95% duration of cell cycle. Interphase. Phase lasts more than 95% duration of cell cycle. Interphase. Phase which is completed in 1 hour. Mitosis. Phase which is completed in 1 hour. Mitosis. Phase starts with nuclear division. Mitosis. Phase starts with nuclear division. Mitosis. Separation of daughter chromosomes called karyokinesis. Separation of daughter chromosomes called karyokinesis. Division of cytoplasm is called cytokinesis. Division of cytoplasm is called cytokinesis. Phase called as resting phase. Interphase. Phase called as resting phase. Interphase. Phase corresponds to the interval between mitosis and initiation of DNA replication. G1 phase. Phase corresponds to the interval between the mitosis and initiation of DNA replication. G1 phase. Phase considered as preparing for cell division. Interphase. Phase considered as preparing for cell division. Interphase. Phase marks the period which DNA synthesis or replication takes place. S phase. The phase marks the period which DNA synthesis or replication takes place. S phase. First stage of karyokinesis. Prophase. First phase of karyokinesis. Prophase. Phase both cell growth and DNA replication occur. Phase both cell growth and DNA replication occur. Interphase. Phase both cell growth and DNA replication occur. Interphase. New DNA molecules formed are not distinct but intertwined. S and G2 phase. New DNA molecules are formed are not distinct but intertwined. S and G2 phase. Initiation of condensation of the chromosomal material. Prophase. Initiation of condensation of chromosomal material. Prophase. The phase DNA replication begins in the nucleus. The phase DNA replication begins in the nucleus. S phase. Initiation of condensation of the chromosomal material. Prophase. Phase cell is metabolically active and continuously grows but does not replicate its DNA. G1 phase. Phase, the cell is metabolically active and continuously grows but does not replicate its DNA. G1 phase. Centrosome undergo duplication. S phase. Centrosome undergo duplication at S phase. The phase, during this time, the amount of DNA per cell doubles and centrosome begin to make towards opposite poles of the cell. The phase, during this time, the amount of DNA per cell doubles, centrosome begins to make toward the opposite poles of the cell. Prophase. Phase, centriole duplicates in the cytoplasm. S phase. The phase, centriole duplicates in the cytoplasm. S phase. Proteins are synthesized in preparation for mitosis while cell growth continues. G2 phase. Proteins are synthesized in preparation for mitosis while cell growth continues. G2 phase. Chromosomal material condenses to form compact chromosome. Prophase. Chromosomal materials condenses to form compact chromosome. The prophase. Complete disintegration of nuclear envelope. Complete disintegration of nuclear envelope. Metaphase. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Prophase. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Prophase. Each centrosome radiates out microtubules called asters. Prophase. Each centrosome radiates out as microtubules called aster. Prophase. Two asters plus spindle fiber plus mitotic apparatus. Prophase. Two asters, spindle fibers, mitotic apparatus. We call prophase. End of this phase, no Golgi complex, endoplasmic reticulum, nucleolus, nuclear envelope. Prophase. End of this phase, no Golgi complex, endoplasmic reticulum, nucleolus, nuclear envelope. Prophase. Chromosomes are spread in the cytoplasm of cell. Metaphase. Chromosomes are spread in the cytoplasm of the cell. Metaphase. Nucleolus, Golgi complex, ER, reform. Telophase. Nucleolus, Golgi complex, endoplasmic reticulum reformed in telophase. By this stage, condensation of the chromosome completed. Metaphase. By this stage, condensation of chromosome completed. Metaphase. At the time of division, the organelles like mitochondria and plastids get distributed between the two daughter cells. Cytoplasmic division. At the time of this division, organelles like mitochondria and plastids get distributed between the two daughter cells. Cytoplasmic division. If karyokinesis occurs, but cytokinesis does not occur, it results multinucleate condition. If karyokinesis occurs, but cytokinesis does not occur, it results multinucleate condition. Chromosome can be observed clearly under microscope. At this phase, chromosomes can be observed clearly under the microscope. Metaphase. Reason for syncytial condition. Karyokinesis does not follow cytokinesis. Syncytial condition reason, karyokinesis does not follow cytokinesis. Morphology of chromosomes easily studied in this phase, metaphase. Morphology of chromosome easily studied under metaphase. Liquid endosperm in coconut is an example for syncytium. Liquid endosperm is in coconut is example for syncytium. Chromosomes made up of two sister chromatids, metaphase. Chromosomes are made up of two sister chromatids at metaphase. Nuclear envelope develops around the chromosome clusters at each pole forming two daughter nuclei. Telophase. Nuclear envelope develops around the chromosome clusters at each pole forming two daughter nuclei. Telophase. Centromere split and chromatids separate. Anaphase. Centromere split and chromatids separate. Anaphase. Chromatids move to opposite pole. Anaphase. Chromatids move to opposite pole. Anaphase. Two sister chromatids held together by centromeres. Metaphase. Two sister chromatids held together by centromeres. Metaphase. Division of nucleus. Karyokinesis. Division of cytoplasm. Cytokinesis. Chromosomes cluster at opposite spindle poles and their identity is lost as discrete element. Telophase. Chromosomes cluster at opposite spindle pole and their identity is lost at discrete elements at telophase. All the chromosomes coming to lie at the equator. Metaphase. Stage, nucleolus, Golgi complex, endoplasmic reticulum, reform. Telophase. The formation of new cell wall begins with the formation of simple precursor called cell plate. Formation of new cell wall begins with the formation of simple precursor cell plate. Kinetochore of chromatid connected to spindle fiber. Metaphase. Kinetochore of chromatid connected to spindle fiber. Metaphase. Cell plate that represent between the two plant adjacent cells. That cell plate. Spindle fiber attached to the kinetochores of chromosome. Metaphase. Spindle fibers attached to the kinetochore of chromosome. Metaphase. The phases of meiosis, reductional division, meiosis, reductional division, meiosis. Stages of prophase 1, leptotin, zygotin, paketin, diplotin, diakinesis in order. Leptotin, zygotin, paketin, diplotin, diakinesis are the stages of prophase 1. Reproductive cell division, meiosis. Reproductive cell division, meiosis. Interkinesis, interphase without S phase. Interkinesis means interphase without S phase. Heterotypic division, meiosis 1 called heterotypic division. Number of haploid cell formed at the end of meiosis 2, 4. Examples of homotypic division or equational division, mitosis. and meiosis 2. Examples of homotypic division or equational division, mitosis and meiosis 2. Largest phase of meiosis 1, prophase 1. Largest phase of meiosis 1, prophase 1, with leptotin, zygotin, paketin, diplotin, diakinesis. Synaptonemal complex is formed at zygotin, synaptonemal complex, zygotin. Synapsis. Synapsis, process of pairing of homologous chromosomes is called synapsis. Process of pairing of homologous chromosome is called synapsis. Bivalent or tetrad. Process of synapsed homologous chromosome, the pair of synapsed homologous chromosome is called bivalent or tetrad. Pair of synapsed homologous chromosome is called bivalent and tetrad. Recombinase. Enzyme involved in crossing over is recombinase. Enzyme involved in crossing over, recombinase. Appearance of recombination nodule in paketin. Appearance of recombination nodule in paketin. Crossing over. Exchange of genetic material between two non-sister chromatids of homologous chromosome is called crossing over. Exchange of genetic material between two non-sister chromatids of homologous chromosome is called crossing over. Synaptonemal complex is composed three thick lines of DNA and protein. Synaptonemal complex is composed of three thick lines of DNA and protein. Dissolution of synaptonemal complex at diplotin. Dissolution of synaptonemal complex or desynapsis is at diplotin stage. The X-shaped structure formed during crossing over, chiasmata. X-shaped structure formed during crossing over, chiasmata. Chiasmata visible at diplotin stage. Chiasmata visible at diplotin stage. Diplotin can last for months or years in oocytes of some vertebrate. Diplotin can last for months or years in oocytes of some vertebrate. Final stage of prophase 1 is diakinesis. Terminalization of chiasmata, diakinesis. Terminalization of chiasmata, diakinesis. In animals, spindle fibers are formed by centrioles. In animals, spindle fibers are formed by centrioles. In plants, spindle fibers are formed by microtubules. Centrioles are absent. In plants, spindle fibers are formed by microtubule. Aster. What is aster? Centriole plus chromosome plus spindle fiber. Centriole plus chromosome plus spindle fiber called aster. Myocyte, cell which undergo meiosis is called myocyte. Cell which undergo meiosis is called myocyte. In algae, myocyte is zygote. In algae, myocyte is zygote. In bryophyte, pteridophyte, flowering plant, myocyte is spore mother cell. In bryophyte, pteridophyte, flowering plant, myocyte is spore mother cell. Diploid condition changed to haploid in meiosis. Diploid condition changed to haploid in meiosis. Haploid changed to diploid in syngamy. Haploid condition changed to diploid in syngamy. Anaphase 1 is characterized by segregation of or disjunction of chromosome. Now phase 1 characterized by segregation or disjunction of chromosome. Meiosis 1 responsible for forming the phase in the life cycle. Haploid phase. Meiosis 1 responsible for formation of haploid phase. Bringing variations in progeny is due to meiosis, recombination. Bringing variations in progeny is due to meiosis. Dyad condition is seen in telophase. Dyad condition is seen in telophase. The stage between meiosis 1 and meiosis 2 is called interkinesis. The stage between meiosis 1 and meiosis 2 is called interkinesis. Meiosis 2 is equal to mitosis. Meiosis 2 is nothing but mitosis. Division of centromere is absent in anaphase 1. Division of centromere is absent in anaphase 1. Meiosis 2 differ from normal mitosis. The two chromatids of chromosome are dissimilar. Daughter cell produced after meiosis 2 are not similar to each other or mother cell. Meiosis 2 differ from mitosis. Two chromatids of chromosome are dissimilar. Daughter cell produced after meiosis 2 are not similar to each other or mother cell. The stage is marked by terminalization of chiasmata, diakinesis. The stage is marked by terminalization of chiasmata, diakinesis. Number of spindle operators formed in meiosis, 3. One in metaphase 1 of meiosis and metaphase 2. Then number of spindle operators formed in meiosis, 3. One in metaphase 1 of meiosis and in metaphase. All spindle fibers attached to chromosome is called spindle operators. Number of spindle operators formed in mitosis, 1. Number of spindle operators formed in mitosis, 1. So, number of spindle operators formed in meiosis, 1 in metaphase 1 of meiosis 1, and in the metaphase 2, 2 are going to form. Then in number of spindle operators formed in mitosis, 1, metaphase of mitosis. Stage, the chromosome become gradually visible under light microscope. Leptotin, here some repetition again because leptotin, zygotin, pacatin, diplotin, diakinesis is a major part. You have a small repetition that all the 5 stages of leptotin, zygotin, pacatin, diplotin, diakinesis. Stage of the chromosome, stage the chromosome become gradually visible under light microscope. Leptotin. Compaction of chromosome continues throughout the stage. Leptotin. During this stage, chromosomes start pairing together, and this process of association called synapsis. Such paired chromosomes are called homologous chromosomes. Zygotin phase. In zygotin phase, chromosomes start pairing together, and process of association we call synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate the chromosome synapsis is accompanied by the formation of complex structure called synaptonemal complex. Synaptonemal complex is formed at zygotin. Electron microscopes of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptonemal complex. In which stage? Zygotin. The complex formed by paired synapsed homologous chromosome is called bivalent or tetrad in zygotin. The synaptonemal complex formed by a pair of synapsed homologous chromosomes is called bivalent or a tetrad in zygotin. Bivalent or tetrad, these are more clearly visible in pacatin. Bivalent or tetrad, more clearly visible in pacatin. The 2 stages of prophase 1 are relatively short-lived compared to the stage pacatin. Leptotin, zygotin are short-lived in compare with pacatin. The 2 stages of prophase 1 are relatively short-lived compared to the stage pacatin, leptotin and zygotin. During this stage, the 4 chromatids of each bivalent chromosome become distinct and clearly appear as tetrad. Pacatin. During this stage, the 4 chromatids of each bivalent chromosome become distinct and clearly appear as tetrad. Pacatin. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosome, pacatin. The stage is characterized by the appearance of recombination nodule and the site at which crossing over occur between non-sister chromatids of the homologous chromosome you'll find in pacatin. Crossing over is the exchange of genetic material between 2 homologous chromosomes, pacatin. Crossing over is the exchange of genetic material between the 2 homologous chromosomes, pacatin. Crossing over leads to the recombination of genetic material on the 2 chromosomes, pacatin. Pacatin stage, crossing over leads to the recombination of genetic material on the 2 chromosomes, pacatin. Recombination between homologous chromosomes is completed by the end of this stage, leaving the chromosomes linked at the sites of crossing over. Pacatin. Recombination between homologous chromosomes is completed by the end of this stage, leaving the chromosomes linked at the sites of crossing over. Pacatin. Beginning of this stage is recognized by the dissolution of the synaptonemal complex. and the tendency of recombined homologous chromosomes of the bivalent to separate from each other except at the sites of crossover. And these X-shaped structures are called chiasmata. It is all happening diplotin stage. In diplotin stage, dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosomes of the bivalent to separate from each other except at the sites of crossover. The X-shaped structure chiasmata will find in diplotin, dissolution of synaptonemal complex and formation of chiasmata are at diplotin stage. Final stage of meiotic prophase 1 is diakinesis. The stage is marked by terminalization of chiasmata, diakinesis. During this phase, chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation, diakinesis. By the end of this phase, the nucleolus disappears and the nuclear envelope also breaks down, diakinesis. Stage of prophase 1 represents transition to metaphase, diakinesis. So, this is all about the divisions of cell division, cell cycle and cell division, the stages of mitosis and meiosis. Excuse me. More to be focused on leptotin, zygotin, pacatin, diplotin, diakinesis and their respective stages. For that, repeated revision is required. Thank you.